Hello, good afternoon, college and teacher. We are the Shower Tanker team formed by Leslie Aremi García Valenzuela, Santi Guillén Gallardo, Matías Ochoa Peña y Roxana Gabriela Samaniego Flores. Then we will present the final project of the aquatic biotechnology subject, products and processes. We will start by defining biomineralization. It's the study of biologically produced materials such as shells, bone, and teeth, and the processes that lead to the formation of these hierarchically structured organic, inorganic composites. The mechanical, optical, and magnetic properties of these materials are exploited by the organisms for a variety of purposes. These properties are often optimized for a given function as compared to the properties of biological materials of similar composition. Material chemists are intrigued by the exceptional control organisms exert over the composition, crystallography, morphology, and materials properties of biominerals and the meal conditions, physiological temperature, pressure, and pH required for them. In recent years, therefore, the field of biomineralization has expanded to include the application of strategies adapted from biology to the production of synthetic materials. Biomineralization is by definition a multidisciplinary field that draws on research from biology, chemistry, geology, material science, and beyond.
Well, now we have the bioaccumulation. Bioaccumulation is the process of the accumulation of chemical substances in living organisms, so that these reach higher concentrations than the concentrations in their environment or in the food. These substances prone the bioaccumulation reach increasing concentrations as the tropical level advances in the food chain. This depending on each substances, this accumulation can occur from abiotic sources like the soil, air, or the water, or other biotics like the living organisms. The main routes of the introduction of a chemical substance is in a living organisms are respiratory, digestive, and interglutamine. And the bioaccumulation is defined as the increase of contaminant concentrations in the aquatic organisms. This is following uptake from the ambiental environment, medium, or the different sources of the exposure contribute to the contaminant bioaccumulation.
Bioaugmentation. Bioaugmentation is the addition of programmed microbial cultures to enhance microbial populations at site to improve contaminant clean up and reduce cleanup time and cost. Bioaugmentation is a bioremediation technology for the treatment of sites impacted with hydrocarbons where the microbiota is increased with the addition of exogenous microorganisms or by the increase of native microorganisms with degradation capabilities of contaminating compounds.
Biostimulation. Biostimulation involves modifying the environment to stimulate existing bacteria capable of bioremediation. This can be done by adding various forms of speed-limiting nutrients and electron acceptors, such as phosphorus, nitrogen, oxygen, or carbon.
Below is showing the comparative table some of the difference between the concepts seen above. For example, Biomineralization is a process by which living organisms promote the precipitation of minerals, either crystalline or amorphous. It occurs through chemical reactions between specific ions or compounds as a result of the metabolic activity of a bacterial organism under certain environmental conditions. Bioaccumulation is active process. Intracellular accumulation of ions requires longer time, performed only by live biomass, need of fresh cultivation medium. Bioaugmentation is the process of adding specific microorganisms to enhance the existing populations and promote biodegradation process. Use exogenous microorganisms. And biostimulation is the process of adding electron acceptors, electron donors, or nutrients to stimulate naturally occurring microbial populations in the contaminated area. Use indigenous microorganisms. As an example of the use of biomineralization, the following article is shown that tells us about that. Much of the recent marine research has been aimed at understanding the effects of anthropogenic induced environmental change on marine biodiversity, particularly for those animals with exoskeletons very calcified, such as corals, mollusks, and urchins. This is because life in your ocean is becoming more challenging for these animals with changes in temperature, pH, and salinity. In the future, it will be more energy expensive to make marine skeletons, and increasingly corrosive seawater conditions are expected to cause these external skeletons to dissolve. However, initial large-scale sensitivity predictions are changing as we understand more about the mechanisms that underpin skeletal production, biomineralization. These studies demonstrate the complexity of the calcification pathways and the cellular response of animals to these altered conditions. Factors include parental condition, phenotype, plasticity, and epigenetics can significantly affect skeleton production and thus future population success.
Now we have a bioaccumulation example. In the article of the herbicide atrazine was used to study mechanisms of bioaccumulation in a freshwater mollusk. The mollusks are Ancylus fluvalis and the fish Coregonus fera. Measurements were performed with the known level and chlorine level atrazine as well as triturate water, and the kinetics of accumulation indicate that atrazine is accumulated via gills, and that or other organisms are contaminated in the blood. The exchange of atrazine was investigated at the equilibrium state of accumulation. The total of residues in organisms is the subject to exchange processes. The rates of atrazine accumulation and the atrazine exchange are similar, and in the investigation of the water balance and revealed that water exchange in organisms is the main mechanism of atrazine accumulation and exchange. Well, the herbicide atrazine was used in the present experiments of the article. This one is one of the most widely used herbicides, both as the selective and as general herbicide in the combined formulations. This is lethal and sublethal concentrations for some aquatic organisms has been determined for the scientist Gulken and Kaschel. Studies of the accumulations of atrazine and other estrizine compounds from the water as well as to some extent by a food chain have been made by the scientist Rogers, and these investigations indicate that only a low level of accumulation of atrazine occurred. The equilibrium concentration began reached within a few hours. And the following contamination in this article was the accumulated atrazine is very rapidly lost from organisms when these are introduced in clean water. The first investigation of atrazine accumulation directly from the water is the contrast of accumulation from the contaminated food indicate that uptake from water was of high significance in the contamination of these organisms. And finally, the investigations are carried out for examining the processes of atrazine accumulation from the water as well as the kinesthetic of accumulations and elimination of the atrazine in the water. This knowledge should lead to the complete understanding of the observed phenomenon of atrazine accumulation in this type of residuals, like the water or the food.
Bioaugmentation example. This article deals with application of Marinobacter hydrocarbon clastic as a bioaugmentation agent for the improved treatment of non-sterile fish wastewater. Marinobacter hydrocarbons clastic is a species of Proteobacteria found in seawater that can degrade hydrocarbons. Well, the wastewater generated by fish processing is characterized by salt concentration similar to or higher than seawater along with high concentration of nutrients, for example, organic carbon and total nitrogen, due to the presence of blood, oil, and fish fabrics. The present study aimed to determine where increasing fish wastewater with Marinabdus sp, Marinobacter hydrocarbon classic, or a consortium of the two halobacteria could successfully improve the elimination of chemical oxygen demand and total nitrogen from fish wastewater. After 9 days of incubation, the bioaugmentation treatment resulted in a significant reduction in chemical oxygen demand, 88%, 91%, and 92% in fish wastewater augmented with Mariner Advance sp. Marinobacter hydrocarbon classic. This study confirmed the efficacy of bioaugmentation in eliminating the chemical demand of oxygen and total nitrogen in saline fish wastewater.
In this work, oxygen depletion was discussed and the use of high-energy ghost electron acceptors for mineralization processes are considered one of the main reasons that limit degradation rates in organic rich aquatic ecosystems. The effect of two different biostimulation oxygen release protocols was investigated using extracellular beta-glucoside activity rates. Sediments rich in organic matter and water were collected from the port of Genoa, Italy. And oxygenation was generated by placing isotopes in the overlying water or by placing oxygen-releasing compounds directly into the sediment in laboratory microorganisms. In the present investigation, it was concluded that the increase in enzymatic activity was significant in the water compartment of the oxygenated microsomes.
Finally, in conclusion, the processes addressed in the presentation generate endless responses with haploid organisms and foods. In the fact of this, we can say that these processes are fundamental in the development and research of various species and organisms.